Rahim. Assalamu alaikum. How are you all? Dear students, once again I welcome you in Pakistan International School TAI's virtual learning system. And this is for session 2020. Dear students, as you know, this is second year class for subject biology. And as you know that in the previous lecture we have finished our chapter number 21 that was all about reproduction. Today we will gonna start another branch of biology, another portion of this book that is about inheritance. That is chapter number 22, inheritance of your book. That is all about genetics. Today we will talk about Mendelian law. So let's start. You know, dear students, that Gregor John Mendel, he was a priest, a monk in Austria, and he studied the inheritance of traits in pea plants. So he developed the laws of inheritance. That's why he called the father of genetics. It means he worked on basics of genetics at the time when the uh, genetics is unknown to the scientists, to the people. He performed several experiments, several crosses, several pollinations among these pea plants. So, as we know that he studied seven of the characteristics or contrasting traits of pea plant, in which there are tall and dwarf, like the height of the plant, seed colors, seed shape, pod shape, and other seven, like including these all seven characteristics he studied. He took first true breeding plant in which like it means the plant that produces all of its offspring of its own phenotype upon self fertilization. So that is called a true breeding plant in which all the genes are same. So let's talk about its first experiment, that is called Mendel's monohybrid cross, or that is also said to be the law of single trait or law of segregation. He performed this experiment on uh, the size of the plant, that is tall and dwarf, and then this Pisum sativum or peas plant, that he got the results. So he observed that their growth of nearly 2 years, up to 2 years, he observed their growth, and that he observed that the tall plants all the tall plants grow later, all tall plants, and the dwarf plants grow into dwarf plants after self fertilization. So then he gave Mendel's first law of experiment. At the time, nobody knows about the genes, so he was the first person who gave the concepts of the gene to um, the human, to like the people living at the time. So his first experiment of inheritance, a law of inheritance that is also said to be law of segregation. So that is about the members of each pair of alleles separate or segregate during the formation of gametes So only one allele can be carried in a single gamete. So we know that during meiosis, gamete formation is done, like sperms and eggs are produced, formed. So while eggs and sperm formation, the genes on the chromosomes are segregated separately, um, like without bonding, without like having the material of the other gene or allele. So they are segregated by themselves, segregated, or they become separate easily. So that's why they can transfer the characteristics to the other organism. So this is said to be as monohybrid cross, or that because that involves only one character, and monohybrid inheritance, a cross which involves a single characteristic determined by one gene. Then he also performed the other on dihybrid inheritance, like a cross involves two pair of alleles determining two characteristics. So in monohybrid cross, what he found out, you can see here, that is the dramatical representation of monohybrid cross. He took parental generation in which he gave the name P1, and from that the first P1 is like the true breeding plant, that is all producing like tall plant, and in P1 the other male or female, the plants are not distinguished here, so the other is totally dwarf. So when the cross or cross pollination is occurred, so what happened in the next plants, some plants are taller, like in F1 generation, if we see its generations, all plants were tall. Again, all plants were tall, but he got the proper results in F2 generation, that is called second filial generation. So in which he observed that by cross pollination of these two plants, he got one tall plant, completely tall plant, the other two also tall plant, and uh, the fourth one, that is the dwarf plant. So he got the ratio here, 1 2 3, 3 tall plant and 1 dwarf plant. So the ratio of monohybrid cross is 3:1 ratio. If we talk about its genotype, let's talk about genotype and phenotype later. Genotype means the genes are present here. So you can see here the genotype of this one, capital T is 1, capital T small t is 2, and small t is 1. So it is genotypic ratio is 1:2:1, and phenotypic ratio is 3:1. So we can just take its the result by making the cross a checkerboard or Punnett square as well. So we can see these are the female gametes, and here we write the male gametes. So in male gametes on F1 generation, where all were tall plant and all were dwarf plant, so we get all tall plant in F1 generation as a result, but they are hybrid, like both of the genes or alleles are present here. Then we will see in F2 generation that male gametes and female gametes both are hybrid. So when we cross them and cross pollination, so what happened, we get completely tall plant 1 and hybrid 2, and that is 1. So this is genotypic 1:2:1 ratio, and phenotypic is 3:1 ratio. So from this we'll get the results. Mendel concluded that inheritance depends on the transfer of hereditary factors. At the time he gave the names, these genes or alleles, as factors from parents to offspring. So there is hereditary factor that determine a particular character. So then each character is controlled by pair of factors. He also found that there are two types of two factors on which whole of this uh, transferring of the characters depends. The factors are passed from generation to generation unaltered. They don't change, but they get separated and then transmit the characters. The factors may be dominant or recessive, and like if the recessive, so there is also the plants production, a different type of plants produced, and dominant also. The factors segregate or separate during gamete formation, and each gamete contained one pair of factors of given characteristics. That was the conclusion of some of the results. Now he got some other conclusions that there are some factors and they are present in the pairs. So he gave the name, the unit of heredity as genes, and the different forms of genes are alleles. So he gave that if the offspring has two dominant alleles, the offspring will appear as dominant trait, and if he has one dominant allele and one recessive, the offspring will also appear as dominant. As we can see here, that green is dominant, so pot color green is dominant, so that's why in hybrid that also appears as green. So in offspring, if two recessive alleles are there, so offspring will show the negative trait. So he just gave the concepts of dominant and recessive alleles also, that the dominant will just write with capital T and recessive will write with a small t, and because they are pure breeding, so they're represented by two alleles, a pair of genes. He also gave this concept, so we write for dominant capital T, and in pure breeding short small t, and in hybrid condition in F1, as we he got in F1 generation, so that is capital T and small t, that is called the hybrid condition of that. So alleles, what are alleles? They each individual carries one copy of the gene for from their mother and one copy of the gene from their father. So one copy from the father, that is also called allele, and one copy from the mother, that is also called allele. So we can see on chromosome number 3 of that pea plant, there is allele for purple flower, and on the other side there's a allele for white flower. So locus for flower color, so that they also represent the locus. Like both of the alleles are pairs, they will be present on the same locus, on the same place. Locus is the place of the gene where they find in the chromosome, so they are present at the same place on both of the homologous pair of chromosomes. So he also gave the concept of genotype and phenotype, that the genotype means which copies the gene of the organisms, and what gene codes for, like it means what gene codes for, like when we just represent the character in genes, so we call it genotype. And if we write the characters in the trait form, so how it looks like or how we can see that trait, so that is phenotype. So here we can see the genotype pp, that is homozygous, and genotype capital P small t, that is heterozygous, means hybrid condition of that, and small p, that is also homozygous. So here we can see that he gave also the concept of genotype by determining homozygous and heterozygous, but these alleles are not present in the form of like one form, but they can be represented in the dominant form as recessive form, as well as in the homozygous and heterozygous form. In homozygous forms, both of the alleles are same. If they are same and they are dominant, that is called dominant homozygous. If they are um, recessive and they are the same genes, they are called homozygous recessive. And if they are dominant and recessive both are present, so that is called heterozygous. Now we will see the second cross or second law of Mendel, that is called dihybrid cross or dihybrid law, because he gave in this two traits are involved. So and Mendel just took the plants of pea plants, and in which he observed two contrasting traits. So that's why it's called di and hybrid also, because in this cross hybrid uh, genotype is also going to be formed at greater ratio. So he took at the time, that is also called independent assortment. Why it is called, we'll say in the next slides. He crossed the organisms with two homozygous dominant and two homozygous recessive genotypes. So, for example, this one capital Y, capital Y, capital R, small y, small y, small r, small r. These are called true breeding plants, in which all the genes are same, all the alleles of the gene is same. So and then he got the result of this type. So we will see what type of results these are. So at the end it produces 9 is to 3 is to 3 is to 1 ratio. What is this, we will see in the next slides. So he gave the law of independent assortment, in which he observed uh, uh, and showed that dominant traits do not always show up together, do not always see green pods and purple flowers in the same plant, because green pods and purple flowers, they are the dominant alleles. So what he observed, that two or more pair of alleles separate independently during the formation of gametes. That is almost the similar to law of segregation, but here two traits are studied. It means there are two pairs of genes are studied in like one organism. So all the alleles which are present in these two contrasting traits, these alleles separate independently. Assort means also separate, so they separate independently from one another. So they do not find any type of changing over there. So equal chances of inheriting blonde hair allele, brown eyes allele, or blonde hair allele, or blue eyes allele. Like there are, what type of characters you are having, they are transferred to the next generation independently. So the traits are inherited separately from each other. Now we will see in the cross, then we can just interpret the results. So first he took pure breeding round plant in round and yellow colored seeds in the pea plant. So for yellow there is capital Y, because yellow is dominant, and then for rounds capital R, and for green and wrinkled seeds of those pea plant, capital small y for green, because that is recessive, and a capital smaller for the wrinkled one. One more important thing is here, when we just combine these, we always write dominant alleles first, we don't write recessive alleles first. So how it will happen, in first p1, then they cross, then gametes are formed, we write gametes in the circles. So because y both are same allele, so we write just one allele here, and from order and we write just one allele here. So because both of these traits, they are just like continue to transmit to the next generation, uh, because both of them are having the different locus, so they are transmitting at the same time. That's why here color and the shape of the seed, they will go side by side in all the organisms. So like every seed or every plant which will produce a seed, the seed will have definite the color or the shape also. So both of the characteristics go side by side, but here we are talking about like the alleles, they form the different combinations. So when we form is a first generation capital Y and small y, capital R, this capital R and small r. So in the first generation we will find a dihybrid organism. It means that in the F1 generation, capital Y come from yellow round plant, and small y come from a green wrinkle plant, as well as the capital R will come from yellow round and wrinkle from green wrinkle. From there small r will come, so it means this is a hybrid, but overall this is yellow round plant. Now when we see its F2 generation, so for F2 generation, now we have to make for cross fertilize, sorry, self fertilize of that. Now if we can see, like the same plant is going to be self fertilized, like a capital Y small y and capital R small r, these two plants are going to be now again fertilized with one another. So when we do this one, first of all we'll make gametes. For gametes, first we will separate them, because in the gametes they will move separately. So how they can, like there from one egg cell we can in meiosis 4 egg cells are formed. So you can just imagine 4 egg cells are formed, how they are formed, like alleles of different, they can combine differently, like capital Y can combine with this R and this R as well, and small y can combine with this capital and small r as well, because both of the traits have to move side by side in the next generation. So gametes of 4 combinations are formed. Now we will take these 4 combinations, and these 4 combinations, male here, male and female will be up, or you can change uh, you can just add female in the side and male gametes up. So when we just cross them, capital Y and capital R, we have to multiply. This is a checkerboard or Punnet's checker square. So we can see this capital Y, like we have to just take this color, this capital Y, capital R will have to multiply or cross with this 1, 2, 3, 4. So this 4 and this 4, 4 fours are 16. So we have to make 16 boxes over there. So first they multiply this one, capital Y, capital Y, capital R, then capital Y, this capital Y, and this capital Y small r. You can see here. So first we write the dominant allele. So dominant is Y. The color. First we have to see the dominant of color, then we have to write dominant of the shape of the seed. So that's why we are writing Y first and R small. So all of them there, if there is capital, it means that is dominant, so we'll write it first, like here capital Y, capital R, and in this now we will see, in this we'll see capital Y small r, you can see here in this box. Then we'll see capital Y small capital R, and then here we can see small y small r. So there will be the combination capital Y small y and capital R smaller. R will be written together and Y will be written together. So now this will just make all the combinations. So next we will just take this one, capital Y small r, so it will multiply with capital Y capital R. So we write capital Y and capital R small r here. Then we multiply capital Y capital small r with capital Y small r here. So we'll get capital Y small r. Then we'll capital Y with this one, capital Y small y and capital R small r we write here, capital Y and small r we write here. Then we will see it's the next one, small y, like small y small r, capital R, it will cross with capital Y small r, capital R, so we write here capital Y small y, capital R, because we have to write dominant. So in Y, capital Y is dominant, so we'll write this one first. So then capital small y capital R with capital Y small r, so I write capital Y small y and capital R small r. Like this we will make all these combinations. So after this we will calculate that the dominant, what is the dominant and what is the recessive. So here you can see in this capital Y, both are dominant, so right, that is yellow, and both are dominant, that is round. So that is the first yellow round. Then if you see capital Y, that is also yellow is dominant, and if the one capital um, letter is present, it means that is dominant. So write the dominant one. So that is capital R, so it means that is also yellow round. If you see here one Y is capital, it means that is dominant, so that will be yellow, and both are round, so that will be said to be round. Then we'll have there is also yellow round and yellow. Now here are two alleles, it means they are wrinkled, so yellow wrinkled. Here we'll get yellow round, yellow wrinkled again, because small r. Here we'll get yellow round also, yellow round also. Now 1 2 3 4 5 6 7 8 and 9 are, now we can see 9 are yellow round, they are dominant. Now we'll see the small letter is, that is recessive, that is uh, green, green, and capital R capital, that is round, green round. Now we'll find out green round in the next one. This is also small y, that is also green, and capital R is present, means green round. And here we are also having green round, so that is 1 2 3, green round is 3. Then we will have this first is yellow, another is round. uh, okay, one yellow round here, one yellow round here, and one yellow uh, okay, yellow round, yellow wrinkled, actually yellow wrinkled. And we are having yellow wrinkled here, number 2. Like we just do this with this one, yellow wrinkled, yellow wrinkled, and the other yellow wrinkled is this one, it means that is also 3, so we'll having another 3. And then we are having just here uh, green wrinkled, so that is only one, because all alleles are we can say just they are recessive. Green round, that is green round 1, green round 2, and green round 3, that is 3. So 9 is to 3 is to 3 is to 1, like 9 or 3 12, 15 and 1 16, all are 16 boxes, so we get 16 the results. Okay, after that, Mendel came to the result that inheritance of each trait is determined by the units A, or factors A, like they are called genes or allele pairs, and an individual inherits one such unit from each parent to the next trait, and a trait may not show up in all in an individual but can still be passed to the next generation, and the genes of each trait segregate themselves during gamete formation. In law of segregation, we are having like law of segregation, we are having 3 is to 1 phenotypic ratio, and in law of independent assortment we get 9 is to 3 is to 3 is to 1 ratio. So Mendel is said to be the father of the genetics because he gave the most important concepts about genetics. But there are certain variations and limitations of the Mendel's law. We will talk about inshallah in the next time. Wish you all the best. Allah Hafiz.